So we've just started our discussion on protein synthesis, and yesterday we looked at the first step of protein synthesis. Remember that synthesis means to build, so we're building up proteins. Now today we're going to focus on the second step, but we want to review really quick about what happened during the first step. Where did we start off in the cell for the first step of protein synthesis? Remember that in step 1, and you might want to do this on a separate piece of paper, on step 1 we talked about transcription. Inside of the cell, we were actually in the nucleus of our eukaryotic cells. Remember that inside of the nucleus we had our DNA, our double helix, and a section of that DNA would open up to reveal a gene. From that gene, we'd actually copy a single-stranded nucleic acid. And what was that called? So double-stranded nucleic acid was DNA. So this was DNA. And single-stranded nucleic acid was what? Well, we actually created mRNA in this process. Then mRNA would leave the nucleus, and today we'll talk about what happens when it heads to the ribosome. This is kind of the big picture of what happened in step 1, transcription of protein synthesis. Remember that if we had a strand of DNA, like AATCCG, then our mRNA would be transcribed, and that would become UUAGGC. Remember that in mRNA we didn't have any T's, we didn't have any thymines, and we had uracils instead. So from the mRNA, now we're going to move to the ribosome inside of the cytoplasm and talk about what happens so we can finally finish building our protein. So in the second step, this is called translation. We basically have our message now, and now we need to read it and make the rest of our protein. This is going to occur in the ribosome. So notice here, transcription has occurred. You have your RNA, and specifically this is mRNA. mRNA will move out of the nucleus through a pore, and it will actually move out to a ribosome. And in this structure, it's really light, but there's actually a ribosome right here that your mRNA is running through. The ribosome is actually going to hold mRNA in place, and it's going to help link those amino acids together. Remember that a chain of amino acids is known as a protein when it finally folds up. So what are the steps to this process? Remember, we're talking about translation. We're talking about the second step of protein synthesis. So in translation, the first step is that your mRNA will enter the ribosome. So this is the picture that you have in your notes. Okay? Here it says it's being exported from the nucleus. So let's add in above this picture that your mRNA is exiting your nucleus. Okay? Once it leaves the nucleus, there's always something called a start codon. Remember that a codon is 3 bases on mRNA. So it's going to join in with the ribosome. Once it joins in on the ribosome, here you can see the ribosome. Here's the mRNA strand, single-stranded nucleic acid. Okay? Make sure we label that mRNA. Then what's going to happen is our tRNA. Here we're seeing our mRNA again. Single-stranded nucleic acid right here. Here are the bases on it. Here are the codons on it. And this shape right here that looks like an upside-down T is actually our transfer RNA, our tRNA. It doesn't always look like a T, but you'll always see it joining in or connecting, looking like it's trying to connect with our mRNA. what it's actually doing is it's actually carrying an amino acid. So the little circle on top of it, that's actually going to be your amino acid. Okay? Let's go ahead and remind ourselves that an amino acid is actually the monomer or building block of proteins. Basically what it's going to do is as it reads the codons on the mRNA, a new tRNA will come, drop off an amino acid, that will leave the ribosome and it'll continue shifting through. So kind of acting like a little conveyor belt and slowly churning out this amino acid chain. So the tRNA's anticodon will bond with the codon on the mRNA. Anticodon is just three bases of your tRNA. Okay, so that's connected there. Then, each new tRNA will transfer its amino acid to the previous one to create your amino acid chain. So again, it's just kind of churning out this chain of amino acids here. So let's add that in. That's your chain of amino acids. And then finally, at the end of the mRNA, you'll reach a stop codon, literally telling the ribosome to stop, you're done making that chain of amino acids. And then your amino acid chain will be released. Let's go back for 1 second. Once this chain is released, this chain of amino acids is released, it will actually be removed from the ribosome or leave the ribosome, and now it will fold up. So released and folds. Okay? Here's our chain of amino acids. Our monomers linked together to form a polymer will fold up that chain of amino acids and actually form our end result, which is our protein. The amino acid chain must fold in order to become a protein. So you can either highlight or underline that, put a star by that. It's really important because we're not a workable protein until we fold into a special shape. Okay? And the last thing that we'll actually need to be able to do is learn how to use this codon chart. It is an mRNA codon chart, and it's used to be able to figure out what amino acid would we get. Inside of this chart, these represent all of the amino acids that can make up a protein. On the outsides, these are the letters that make up the codon. So if you notice, here's our first letter going down this row. Here's our second letter going in the middle, UCAG. And here's our third letter going down the sides. So let's work backwards a little bit. If I told you to figure out what code or what codon makes up this phenylalanine, the first box on your upper left-hand corner, what letters would you tell me would code for that amino acid? Here's the amino acid, so my amino acid is phenylalanine. I'm going to shorten that, phe. And you tell me what three letters would make that up. So work backwards with this chart. Hopefully, you looked at the first letter. Here's U. Second letter, again, this is going to be U. And my third letter in this case will also be a U. Okay? So this actually codes or makes up my mRNA that codes for this amino acid, phenylalanine. Now let's try another one. If for the second one you have, let's say this stop codon, okay, 3 down on the right, what amino acids, what mRNA sequence codon would code for this stop? So use the chart again. So if my amino acid is stop, this specific stop, what letter letters would make that up? Well, the first letter again would be in from this box. I bring my finger across. Here's my stop. So that means my first letter for my mRNA is U. Then I look at my second letter. I move down. Here's G. And then in my third letter, I found A. So UGA would be the mRNA that codes for this specific stop amino acid. Then what you have next at the end of your notes is your summary box. In your summary box, it says to use that mRNA sequence that you transcribed on the previous page. You're actually just going to use that on the previous page. You're going to use that sequence starting with AUG and tell me what amino acids would make that up. So let's go ahead and start that. If I have AUG, what would the amino acid be for that? I would draw a dash right in between here. So if we look, first letter A, second letter U, so I'm down in this section of the codon chart, and I find G, I bring my finger together, and I found start or methionine, okay? So you can actually write start or met. I would just write the first three letters. And then the next set is GUG. Okay? Figure out the rest of these amino acids to build that protein. And that's all for today.